For years, they had poured everything into this. Late nights, relentless focus, unwavering determination. Every experiment, every failure, and every small step forward, all leading to this one hope that their drug could change lives. The entire team had prayed for this moment. And then, one ordinary morning, he walked into the lab just like any other day, but this time, something was different. There, on his desk, a letter. It's the FDA one, and he directly knew what it concerns. His heart raced as he tore it open. And then, he wrote that his drug is approved. The developed drug by his colleague, which is designed and synthesized by him, is now officially approved to treat patients with ATTR-CM disease. After years of struggle, they done it. Hands trembling, he grabbed his phone and called the team to tell them the big news. This was it. Their years of sacrifice had finally paid off. Their names would now be etched into medical history, forever remembered for the lives they were about to save. This was more than just success. This was a revolution. Hello everyone, welcome to Altan's Pharmaceutical Journey Show, your guide to the evolution of the pharmaceutical field through history and its latest innovation of today. I'm your host, Abdulani Abdali. In today's podcast, we'll be discussing the Acoramidis medicine. Stay tuned to discover the story of this latest drug development approved on November 22 of last year and how it could save lots of patients' lives with its efficient characteristics. Wishing you an enjoyable listening experience. Originally developed by Isabella Graef and Dr, a former faculty member at the Stanford School of Medicine, and designed and synthesized by Mamun Alhamadsheh, PhD, a former chemist at Stanford University of Pacific. With the collaboration of Ron Waitless, MD Professor of Medicine, and Michael Leitke, MD Associate Professor of Medicine. Also played key roles collaborators during the early research on the drug Acoramidis, previously known as the Alhamadsheh Graef Molecule 10, AG10, received an approval for the treatment of ATTR-CM disease on November 22, 2024. People who are diagnosed with ATTR-CM is the abbreviation of the disease Transthyretin Amyloid Cardiomyopathy. Those patients experience a buildup of misfolded protein called transthyretin on their heart, causing stiffness, which means rigidity or lack of flexibility that prevents the heart from beating properly. This new drug called Acoramidis playing its role as a transthyretin stabilizer, which stops the protein from misfolding and aggregating the heart. So before deeping inside these disease details and the details of this latest developed drug, let's get in the story of its development path followed by the developer's scientific researchers team. First of all, this drug is not the first one developed for treating the ATTR-CM. So, dear listeners, when I say ATTR-CM, just know it is the transthyretin-related amyloidosis cardiomyopathy, which is the disease that Acoramidis treats. Acoramidis is not the first one. Tafamidis came to the world as transthyretin protein stabilizer before Acoramidis. However, to distinguish the difference and see the impact changement that Acoramidis brought, you have to know that the ATTR is a rare and very complicated chronic disease. It is exactly a mutation-based genetic disorder due to the accumulation of unstable transthyretin protein. and presents with symptoms of congestive heart failure and numerous extra cardiac symptoms like carpal tunnel syndrome and neuropathy. There is two types of this disease. The first one is the hereditary ATTR-CM, which is called the familial type. This one is so complicated and there is no clear research as results about it. The second one is the wild type, which appears with the age advancement. Acoramidis have a very detailed mechanism of action for the ATTR-CM, and with its precision mechanism of action came the difference between it and the first drug tafamidis on the efficiency. Before we completely get in the story, you should be informed that untreated patients with ATTR-CM have median survival of 2 years and 6 months until 3 years and 7 months, dependent on disease stage. So this illness is almost dangerous as cancer. The story began when the research team had the insight that achieving complete stabilization of both normal and mutant TTR required a molecule with a novel binding mechanism, one that, unlike existing other stabilizers, also interacts with the bottom of the T4 binding pockets of TTR. To understand generally this process, I left you in the description a link to the drive that contains an image of the TTR protein form. Okay, so you see and you will know exactly what is the T4 binding pocket. So the research team during the research are trying to find a stabilizer that fits like a hand in a glove into this pocket. The pocket is the T4 binding pocket of the TTR protein. At Stanford, the team conducted the first high-throughput screen for TTR ligands, uncovering novel and structurally diverse compounds. Using structural insights from the co-crystal structure of this ligand with TTR, Dr. Alhamadsheh designed and synthesized AG10, Acoramidis, a compound that fits the binding pocket with remarkable precision and stabilizes transthyretin protein in patients with ATTR-CM by mimicking the protective effect of the rare T119M mutation. The T119M mutation is just the genetic disorder of the TTR protein that leads to its destabilization and then creating the ATTR-CM disease. Let's continue the story. In 2012, Dr. Graef and Dr. Alhamadsheh became part of the Stanford Medicine SPARK program for rare heart conditions, a program that played a key role in supporting the development of this drug. After that, Dr. Alhamadsheh and Dr. Graef co-founded Eidos Therapeutic in 2013. Eidos' public offering in 2018 accelerated Acoramidis' progress. The 2021 year had seen a merger with Bridge Biopharma. For those who don't know, Bridge Biopharma is a new type of biopharmaceutical company founded to discover, create, test, and deliver transformative medicines to treat patients who suffer from genetic disease. Its team of experienced drug discoverers, developers, and innovators are committed to applying advances in genetic medicine to help patients as quickly as possible. So, researchers from Eidos Therapeutics and Bridge Biopharma contributed to the clinical development of this drug and ensured the successful completion of clinical trials that demonstrated an improved efficacy compared with those already available, like tafamidis. Patients experienced better survival rates and quality of life. And with these clinical trials results, the Acoramidis drug was established as best-in-class therapy of ATTR-CM to get its approval by the FDA on the end of 2024. Now I let you listen to what Dr. Mamun Alhamadsheh said concerning this successful project of Acoramidis drug development.
I'm currently a professor of pharmaceutical sciences at the University of the Pacific, that's in California, Northern California, San Francisco. But I'm also a co-founder of a biotechnology company in San Francisco that focuses on drug development for heart disease drugs. To be honest with you, the rigorous academics that I received while doing my PhD at Toledo, I was there from 1999 to 2004. I did my PhD there. I think the rigorous academics were very important for providing me with the foundational knowledge to continue in the drug discovery. You know, I was in the pharmaceutical science mid-chem department. But also, I believe the relationships I built with my mentors and the way that they taught me and mentored me and nurtured me were very crucial in advancing my academic career down the line, you know, as we're gonna discuss shortly. During my pharmacy education, that was in my country, home country of Jordan, and then I came to Toledo. I continued in medicinal chemistry. I fell in love with organic chemistry. And this combines with my passion for research and also the love for teaching, made my career choice in academia kind of very obvious. Then I went into the academic career, and we were very fortunate to work on a very exciting project that resulted in a molecule that's called AG10, where basically, you know, is a drug candidate that we hope that will be approved by the FDA by the end of this year.
Dr. Graef said that this drug represents a beacon of hope for patients with ATTR-CM and commented, "This success is the result of years of dedication, collaboration, and shared commitment to improving patients' lives. I'm truly grateful that Dr. Alhamadsheh and I have been able to contribute to a therapy that can make a meaningful difference for those affected. We both hope this achievement inspires the scientific community, particularly young scientists, by showing what can be accomplished through perseverance and teamwork. It demonstrates the importance of sustained effort and commitment in tackling complex challenges. We are also grateful for Spark's mentoring and guidance, which was so important for us into developing AG10 Acoramidis." That was Dr. Graef says. Now, after telling this amazing story of Acoramidis development, let's get into the details to know exactly what this dangerous and rare heart disease called ATTR-CM is. How does the Acoramidis drug play its role exactly in treating this disease? How were the clinical trials conducted to confirm its efficacy? How is Acoramidis prescribed and what are its pharmacological properties? We will have answers for all these questions in the next parts of this episode. So, let's start with understanding the disease. Transthyretin-mediated amyloidosis can be inherited as an autosomal dominant trait. To explain this for those who don't know, autosomal means the gene is on a regular chromosome, not the sex chromosome X or Y, and dominant means just one bad copy of the gene from your mom or your dad is enough to cause the trait. So, this autosomal dominant trait is caused by pathogenic variant mutations in the TTR gene or by deposition of wild-type TTR protein, ATTR-WT. First, the DNA is transcribed to RNA, which is the messenger RNA. Then the mRNA is expressed into protein through the combination of different amino acids. If the gene contains a mutation, it produces an unstable or inactive protein. That is the mechanism that happens in this case. The produced TTR protein is destabilized and dissociates into unstable monomers that deposit as amyloid fibrils in the myocardium. This can lead to cardiomyopathy and polyneuropathy. Treatments depends on distinguishing between ATTR-V, which is the hereditary one, and ATTR-WT, which is the wild type one, to determine how it can be treated. ATTR is the general one, but our subject of the study is the Transthyretin Amyloid Cardiomyopathy, ATTR-CM, which is what our today's episode's drug treats. So, ATTR-CM is an underdiagnosed chronic disease associated with progressive heart failure. It results in impaired quality of life, repeated hospitalization, and premature death. Acoramidis is a selective oral transthyretin stabilizer that prevents its dissociation into unstable monomers that deposit as amyloid fibrils in the myocardium. Acoramidis was well tolerated and showed clinical efficacy in improving the primary endpoint, a hierarchical combination of all-cause mortality, cardiovascular-related hospitalization, and N-terminal pro-B-type natriuretic peptide level. To keep this last one simple for you, N-terminal pro-B-type natriuretic peptide, it is just a blood test that tells doctors how hard your heart is working and if it may be failing. A high level means your heart is under significant stress. With this general information about this complicated rare disease, we will get in the clinical trials did on Acoramidis and what was the results this one gave to see exactly its efficacy level. The objective of the clinical trials is to characterize the efficacy of Acoramidis on ACM, amyloid cardiomyopathy, and CVH, cardiovascular hospitalization. The used methods for the study are the following. Participants with ATTR-CM were randomized with a ratio of 2:1 to receive 800 mg of acoramidis hydrochloride twice daily or placebo for 3 months. To explain you this randomization of 2:1 in these clinical trials, it's just there is two groups. The first group gonna be tested with real acoramidis medicine. The second one will take a placebo. So the first group A and the second group B. When they will choose people, the two first people, they add them to the group A, the group that's gonna be tested with real acoramidis medicine. The third one gonna be added to the group B, which is the group that's gonna be tested with a placebo. Then after the third one, they will take two others to add them to the group A. Then they will take one to add them to the group B. So this is generally the randomization with a ratio of 2:1. Now, efficacy analyses were conducted in the modified intention to treat population. That means that they've chosen participants with baseline estimated glomerular filtration equal or more than 30 milliliter per minute on 173 meter cube. The glomerular filtration is a blood test that measures how good the kidneys filters waste from the blood. So the chosen participant for these clinical trials must show a result of 3 milliliter or more of filtered blood per minute to ensure trial participants have kidney healthy enough to process the drug safely. CVH and the first composite of ACM were plotted by using Kaplan-Meier curves and summarized with a stratified Cox proportional hazard model. Subgroup analyses were conducted for the composite of ACM or first CVH. The composite of ACM or first CVH is a primary endpoint in ATTR-CM trials that combines two critical outcomes. The first one is ACM, which means death from any cause. And the second one is CVH, which means the initial hospitalization due to the cardiovascular event like heart failure exacerbation. Thus, it is a validated measure of disease-modifying effects in ATTR-CM. Now, we arrive to the important point of this study, results. So, from the 632 participants randomized to treatment, 611 were included in efficacy analysis, means 97%. 409 received Acoramidis and 202 received placebo. So compared with the placebo, Acoramidis reduced the occurrence of ACM or first CVH. The annualized frequency of CVH was reduced with Acoramidis compared with placebo, and the efficacy on the composite of ACM or first CVH was consistent across subgroups. Acoramidis was well tolerated with no safety alerts of potential clinical concern is identified. As a conclusion for this clinical trial, we can say that in patients with transthyretin amyloid cardiomyopathy, the TTR stabilizer had benefits over placebo on the primary endpoint, including mortality, morbidity, and function. This proves the efficacy of this newly developed drug. However, the clinical trials is not complete and does not confirm the hypothesis. Why? Definitively because it has limitations, such as most patients having wild-type ATTR-CM, limiting generalizability to hereditary variants. Also, the majority of patients were white and male, restricting demographic applicability. Beside this study, the American Heart Association has issued guidelines on cardiac amyloidosis management, but did not discuss Acoramidis' place in therapy, as the guidelines predate its approval. In recent analysis, Acoramidis received a moderate certainty recommendation for ATTR-CM treatment based on improved clinical outcomes with uncertain mortality benefit. Means that Acoramidis can delay death but not prevent it indefinitely. We arrive to the last part of this episode. After we've seen the story of its development, the role of this drug and the clinical trials done to prove its efficacy, let's see how this drug is taken, the dosage, and its contraindication. First of all, administration. Acoramidis is administered orally with or without food and with whole tablets swallowing without cutting or crushing. For the dosage, it's 2 tablets of 365 mg daily. There is no specific dosage recommendation for hepatic and renal impairment and no dosage adjustment needed for patients equal or over than 65 years of age. Concerning the contraindication, there is no contraindications and no sufficient data for pregnancy to determine risks of major birth defects, miscarriage, or adverse maternal or fetal outcomes. This medicine is not studied in pediatric patients less than 18 years old, so we cannot say anything about that category. However, the common adverse effects that come from this drug is diarrhea and upper abdominal pain. Now, besides the pharmacological properties of Acoramidis, there is another factor to consider: drug interactions. As you know, sometimes it occurs when your doctor prescribes a medicine for you and asks if you take medication from other therapeutic classes. For example, if he asks you whether you take drugs for a hypertension, and you say yes with specifying the drug, he may instruct you to take, for example, either 2 hours before or 2 hours after the prescribed drug, or he advises you avoiding certain foods during treatments. Well, these instructions share a common objective, which is avoiding food and drug interactions. This means some food or medications can create an obstacle for the given drug to exert its therapeutic effects. Therefore, let's examine drug interactions specific to Acoramidis and medications you should avoid taking them concomitantly with Acoramidis. We start with drugs affected or metabolized by hepatic microsomal enzymes. The first one is CYP2C9 substrates like non-steroidal anti-inflammatory drugs and oral anti-diabetic agents. They are drugs or compounds that are metabolized by the CYP2C9 enzyme, a member of the cytochrome P450 family. This enzyme plays a crucial role in the oxidation and breakdown of various substances inside the liver. Acoramidis may increase concentrations of the substance drug. Monitor patients for signs of toxicity when used concomitantly. So you shouldn't take Acoramidis concomitantly with drugs like anti-inflammatory ones and oral anti-diabetic agents. The second enzyme is CYP3A, like benzodiazepine, calcium channel blockers, macrolide antibiotics, and steroids. They are drugs or compounds which are metabolized by the CYP3A enzyme. The activity of this last one can be influenced by inhibitors, which slow the metabolism, and inducers, which accelerate metabolism. This category of drugs can reduce the exposure of Acoramidis, the reason why we should avoid concomitant use. Now, drugs affecting or affected by transport system. UGT inducers. They are compounds that increase the activity of UDP glycoronosyl transferase enzymes, which are crucial for phase 2 drug metabolism. These enzymes facilitate glycuronidation, a process that enhances drug elimination by making substances more water-soluble. They may reduce Acoramidis exposure, so avoid concomitant use. After this drug is taken while respecting those pharmacological properties, it will follow a full chain inside the body from absorption until its elimination. Now we are going to discover together the process of this drug inside our body. First thing that happens to a drug once it is inside our body is its absorption into the bloodstream, after which its concentration rises in the plasma. For Acoramidis, the peak plasma concentration is achieved within 1 hour, meaning the drug is quickly absorbed. Regarding food effects, Acoramidis pharmacokinetics are not affected by high-fat meals and are not significantly affected by age, race, ethnicity, sex, or renal impairment. After absorption, the drug will be distributed to the therapeutic targets to exert its effect. Acoramidis is 96% protein bound, meaning 96% of circulating Acoramidis molecules are bound to TTR, while 4% remain free and bound in plasma. The last pharmacokinetic phase is elimination. Acoramidis is eliminated 32% in feces and 68% in urines. Another key pharmacokinetic parameter is half-life, which is the required time for drug concentration in the body to decrease by 50%. The half-life of Acoramidis is 6 hours. As a general conclusion, we can say that Acoramidis represent groundbreaking advancement in the treatment of transthyretin amyloid cardiomyopathy, offering hope to patients with this rare and life-threatening disease. As a selective transthyretin stabilizer, it addresses the root cause of ATTR-CM by preventing misfolded TTR from forming amyloid fibrils in the heart, thereby improving survival rates and reducing cardiovascular hospitalization. Finally, Acoramidis exemplifies the power of translational science from Stanford's labs to FDA approval and underscore the importance of collaboration between researchers, clinicians, and biopharma innovators. While challenges like cost and long-term outcomes persist, this drug marks a pivotal step toward transforming ATTR-CM from a fatal condition into a manageable chronic disease. For patients, it's more than medicine, it's a lifeline. For science, it is a blueprint for tackling rare disease with precision. The journey of Acoramidis is far from over, but its legacy as a best-in-class therapy is firmly established. That brings us to the end of this Acoramidis drug episode. Thank you for joining us on this deep dive into Acoramidis, and we hope you enjoyed it. We must inform you that the story told in the beginning of this episode is invented by us, and there is no evidence that it was like that in real life. Unlike the shared information about Acoramidis, which are sourced from official and credible bibliographic resources. Below, we'll leave you the link that directs you to the published article in our bibliographic resources in case you wish to deep your search about Acoramidis. If you have any questions, feel free to share them in the comments. Don't forget to subscribe to this podcast and stay tuned for more breakthrough on the next episode of Alsan's Pharmaceutical Journey Show. Abdulani Abdullah was with you. See you in the next episode, dear listeners, and take care of yourselves. Goodbye.